biologist. Those are plants. But also to tell you a little bit about what we're working on, but also a little bit about my journey to where I am now. And I actually came to plant biology quite late in my career and in my life. And so for those of you who are thinking about switching, either because you want to switch fields or you have to switch fields, perhaps some of what I'm going to talk about will have some resonance. So another title for this might be the development of a developmental biologist. So that's really what I am. That's what I consider myself. I'm interested in how you go from a single fertilized egg into something that is wonderful and complex, and all of the differences and asymmetries that come along with making all of those different cells. So I started in my undergraduate, I was a biochemistry and molecular biology major. When I started graduate school, I wasn't entirely sure what I was going to do, but in the American system, you get to sample a couple of labs. And so when I was a graduate student, I ended up at the University of Colorado, and I, somewhat to my surprise, absolutely fell in love with this worm. So this is a C. elegans. It's a nematode. It's been used for developmental genetics for a number of years. And I think the reason that I fell in love with this had a lot to do with the community of the people who were working on this. They were excited about things. It was a time when we had no multicellular organisms that had genome sequences, which is kind of amazing that it wasn't that long ago. So this was going to be the first organism with a sequenced genome. There had been previous people who had worked out how every cell in that worm, the lineage of every cell in that worm. So we knew a lot about how to make that. And then also coming online were tools to be able to watch development live in action. So GFP was being put into these animals, and so you could watch how development unfurled. And so one of the things that I just absolutely loved was this question about how do you take a cell and make it into two different cells that are going to be different in their size and what they contain and what they'll become. And so this is actually a movie that was made after I finished my PhD by a different group, by Nate Goring. But just to kind of illustrate the thing that I found absolutely fascinating was to look at the beginning of life, really. Hopefully this movie plays. And what you'll see here is it's fertilization. We've got nuclei coming together. We see proteins that are on one side or the other. The cells will divide, and we'll end up getting two cells that are different from one another. One of them is going to make the germline, and one of them will make everything else. And so to be able to watch that happen was totally thrilling. Now, my own PhD was challenging. I didn't actually succeed in answering the question that I had hoped to answer when I started. Although, interestingly, I actually met, where's Sundar? I actually met the person who did solve my PhD problem. So he actually answered the question that I'd been trying for 20 years to answer. He did that in his PhD, so that's really amazing. But I felt at the end of my PhD that I needed to move on to something different. So I was thinking, as many people do when they make a transition, what's important to me? What should I do next? And so one of the things about worms is that there are 959 cells, and there are 959 cells no matter what you do to that worm under normal development. But most of life is not that consistent. And so I became interested in flexibility. So maybe that was flexibility under different environmental conditions. Maybe that's our own flexibility. If we feed us more, we grow more. And so I found that worms were a little restricted. They weren't so representative of the life that I saw around me. The other thing had a lot to do maybe with my own trajectory, where I'd made a lot of mistakes, and I wanted to believe that from a developmental point of view, it was possible to go around mistakes or repair mistakes or go beyond those, and that wasn't really what worms were doing. And then, like many people, how can I work on something that matters? And by matters to the scientific community, matters to the world. How do you choose those kinds of problems? So in thinking about all of those things, I ended up switching from worms and starting a postdoc in plants. So I don't work on these, but these are important plants. They are very visible kinds of plants, but they're also a great illustration about how most life is a bit more flexible and adaptable than a worm. So this is growing out somewhere in California. If you grow the same genetically identical organism in your living room, you can see that there's a lot of compensation. Development of these organisms depends very much on the environment. They're also very able to overcome mistakes. You can chop off a limb and it'll regrow or regrow something else. But I didn't really realistically work on trees, so I went back to those roots of working on genetic model organisms. So for plants, Arabidopsis is that model organism, and I still couldn't let go of that love that I had for being able to watch development, having these beautiful movies where you could really get an idea of what was going on in that organism. And so I worked on Arabidopsis, but I worked on something that was about as close to a worm as you could imagine. So I started to work on the surface of the leaves, where there's development of, it's a very simple kind of thing. You make, the surface of the leaf is waterproofed so that water doesn't leak out, and that's because of these cells that are interdigitated. But plants are also communicating with the environment, and they're actually taking carbon dioxide in from the atmosphere for photosynthesis. And so one of these cells, one of these pairs of cells, is called a stoma, and this is what I ended up working on. So a stoma is a valve. It's two cells. They open and close, and when they open and close, that changes the ability of the plants to take in carbon dioxide from the atmosphere that goes into the plant. It's fixed in photosynthesis, makes food. And on the other hand, water that's been in the soil that's gone up through the plant is released through the plant, as well as oxygen, a byproduct of photosynthesis, and that creates the air that we need to breathe. So I quite like these, and they're important. And it's an example of something, these are minuscule. You need a microscope to see them. But if you think about their collective activity, so the millions of stomata on the millions of leaves of the millions of hectares, they're actually changing our entire climate. So in terms of something that matters, these little guys do matter. And the way I think I can illustrate that best is actually show you a satellite view. So I'm going to walk you through, I'll show this to you in a movie in a moment, but this is a view from two satellites, one in the atmosphere that's measuring carbon dioxide levels, so that's in orange, and the other is monitoring vegetation on the ground, so that's in green. And if you really want to get an idea of what these plants are capable of doing, you can see this in these kinds of global scales. So what I'm going to show you when the movie starts, you're going to basically see the Earth breathing. So I'll let it go for a year, and then I'll talk over it. Okay, so what we're watching is, so in December and January over the northern part of the world, we're heating houses, so carbon dioxide's in the air, there's not much vegetation, but as soon as that vegetation is super active at the end of the summer, all of that carbon gets sucked in. And so basically we're watching our human production of carbon dioxide and plants fixing that problem by taking it back into the biosphere. And that's all through these little pores that I was working on. Okay, so they became, I thought, fairly important. But the question was, I was still a developmental biologist. I wasn't going to become a global climate scientist. So how could I take these and mold them back into the kinds of questions that were interesting to me? And so it turns out the development of these, so they're important when they're formed, but the development actually distills many of the kinds of themes that we see in development in all organisms. So to make these, there's an asymmetric division, just like there was in C. elegans, an asymmetric division that created a cell but then behaved like a stem cell, so that was flexibility. It could divide more times. The more times it divided, the bigger the leaf was, the more stomata there were. And then a transition that allowed these cells to become two different cell types. So starting with that kind of biology, then my program as a postdoc and now with my own group kind of took a couple of different tints. So we became interested in these cells. We became interested in them at the larger scale. So what's the physiology and diversity of plants that matter for our food and our climate? And then this question that was still in the back of my head. So to get at these, I wanted to take a step back and think about what were the strategies that really became important for allowing me and my lab to do the science that we wanted to do. And so I'd say the first thing was really going into something new, embracing new technology to do that, but also some old technology, and really laying out the question in a way that was interesting and accessible, and that an audience that might not normally look at plants might be able to look at them in a different way. Then there are a number of things, I'm actually going to come back to these in a few slides, about some things that I learned along the way. And then finally, something to echo what Ron had said yesterday, developing a supportive lab culture really enabled all of these things to happen. So embracing this new technology and getting foundational work, many of you might end up in new fields, and it's important to lay the groundwork. So for us, that meant making leaves much more exciting than probably most of you think that they are. So rather than just seeing a leaf that doesn't seem to be doing much, by creating new reporters and imaging setups, we were able to take something like this, color code it, so we have all of the cells outlined. We've got green marking stem cells, we've got blue marking the pre-stomata, and develop the ability to capture this over time. So this is a leaf growing. You can see these things bouncing on and off, the stem cells going on and off. And if you zoom in, you really get a chance to watch what's happening at the surface of these plants as they're developing. And so I just really kind of fell in love with this. You know, what's going on with this cell where it's a stem cell? For a moment, both of its daughters are the same thing, but then they resolve it. So you've got an error correction. So they both can't be stem cells, only one can. How do they solve that problem? How do they communicate? So we laid the groundwork there and then began, as molecular geneticists, to identify some of the genes that were important for that. I'll go through this fairly quickly, but the things you were actually watching, the green and the blue, those are reporters of transcription factors that are required for cells to undergo asymmetric and stem cell-like divisions and then to stop undergoing asymmetric stem cell-like divisions. And so those would be color-coded there. More molecular genetics identified that these were regulated by signaling pathways that are familiar to animal biologists, MAP kinase and GSK3 beta. And we also found other things that might be familiar to you as animal biologists that had to do with cell cycle. And that initial thing allowed us to start to ask questions about how do environmental or systemic inputs feed into development, and also fate and memory and reprogramming. Because plants are really good at, you chop off a limb and it'll regrow it, they're actually a great model for understanding regeneration. So one of the things that happened in doing this was that we actually realized that many of these molecules and mechanisms were quite conserved between plants and animals. It's not something that I necessarily expected, in part because the last time that plants and animals shared a common ancestor, they were a single cell. And so everything about multicellularity, everything about development was actually convergently developed. And so the fact that they came up with the same solution, I think, tells us something quite interesting about how you create stem cell-like lineages and how you go through development. So that became quite interesting. And then also conservation allowed us to go from something that was really a model into other plants, because it turns out that these transcription factors, you can find them in other plants and they're used for the same kinds of roles. So, and then finally this asymmetry, this sort of love, this thwarted love of my PhD, where I really was excited about it but never got anywhere. I thought I would never get to this because all of the molecules that actually are important for animal asymmetry, in contrast to the other things that I told you about, the molecules for asymmetry are actually not in plants. But through some hard work by a number of people in the lab, we were able to actually find things that are asymmetrically localized. I'm going to show you a movie here in a second where you can watch. This is a fun thing to watch. So these proteins show up, go over there, the cell divides asymmetrically, and every time they go through a stem cell division, these kinds of proteins mark the two different cells. This is completely a novel, new protein, new kind of mechanism. We call it basal. We had no idea how it worked, and after 10 years of really kind of beating our heads against the wall, I think we've kind of come to the other end of actually finding something that's in parallel to what we know about in animals that allows cells to become different. So those are the kind of things that we've been really focused on, but let me go back to some of those big picture things. So how does my approach to these maybe parallel what you might be doing in your own groups? So this was about embracing old and new technology. But I think it became important to develop a scientific niche. So I think for everyone here, you need to be known for something, and what you're known for is a product of your history and what you're looking for in the future. And so there were a number of things that became, I think, important for us to go forward with this. So one of the things, I say, so I'm going to give you some advice. So advice number one is becoming the missing link. So a lot of what I do, and actually really enjoy doing, is being kind of between communities. So a lot of times I'm the plant biology speaker at an animal medical or animal developmental meeting. And sometimes I'm the molecular biologist at an ecology meeting. And so if you have the interest to do that, it's actually a really great position to be in. You can link two fields together and learn from both of those. And with one experiment, you can actually end up having a continuum of things. So that you can be a missing link in a number of different ways. So in the case of asymmetry, this is something that it was a connection to my past. Because I knew people in animal fields that were interested in cell biology and the techniques in order to visualize things, I could tap into their expertise and bring that to a new field. In terms of physiology and diversity, we actually do, believe it or not, a little bit of climate modeling. And there's no way that I have any training in that, but we're able to convince those folks that we had some interesting ways of using genetics to test ideas, and they could go and test them. And so if you can position yourself in those ways, that's quite powerful. The other thing I'd say is look at the classic studies. It's something that, particularly in the internet age, we don't like to go to libraries anymore. But pretty much everything that I think is a really exciting question, E.B. Wilson wrote in a book in 1898. So some of the really fundamental questions have been there for a long time, and a lot of data is out there. And if you can tap into that, you actually get a head start. So in my case, this was because stomata, they're on the surface of plants. When plants fossilize, you can actually see them. They're being used for taxonomy. There's a great deal of literature out there. This is actually from Delhi, where people have described various things. And so digging through there, you have this treasure trove of interesting biology that you can come back to. And this is not something that's unique to me. There are a number of other people that have really been successful in this kind of approach. To just give two examples, Bob Goldstein, who's a C. elegans biologist, among other things, he reads really old papers. So this is something from 1903. He looks at this, he looks at what's been discovered, says, I've got a new tool, I've got a new trick to monitor these things. Let me bring that into the 21st century, and has really, with C. elegans, done some really nice work. On another scale, Hopi Hoekstra, who is an evolutionary biologist and works on behavior, she once told me, so she works on mice, is that all of the projects that she starts, she first goes and asks, did someone do field work? Did someone find a population of mice that, and work out all of their genetics and heredity and find something interesting, and can she go back and revisit that? Because a lot of the groundwork has already been done. She can just revisit that with where they got blocked at some point, and she's got new technology. So my third piece of advice would be take advantage of the resources that are unique to where you are. So for us, sometimes we live on a campus and we've got a lot of undergraduates that we need to keep busy. And so sometimes we've sent them out and they've brought back, this is a plant that does 2 different types of photosynthesis, and that's actually turned out to be interesting. This is from a course that I did on algae, which I knew nothing about till I got up there and stood and talked about it. And we found some things that we're going back to because they've got really interesting asymmetries. And again, we're not the only ones who've done this. In fact, there's a great example. Some of you might be colleagues with Utpal Nath. So Utpal was a developmental biologist of plants. He did his postdoc in England and studied how leaves grow. But he used leaves that were on plants that were in England. And when he returned to India, he actually revisited those things in these really, really elegant studies, where essentially he said, you know, we used to think this is how leaves grow, but no, this is how a leaf grows. And figured out by using indigenous plants here that there's a lot more diversity in the way that leaves grow. So there's a fundamental changing of the way we thought about biology by using things that were probably in his backyard. And you can see that with biotechnology. So Luis Herrera has done some of this great work. So he's in Mexico. He went out into these areas that have all sorts of, they're hard to live in areas. And the microbes that are in these areas have found amazing ways, amazing biochemical pathways in order to strip what they need from the land. And so by looking at how they acquire phosphate or phosphorus, he actually found that there was a trick that you could do to make plants able to use phosphite instead of phosphate in order to grow, and that allows, so this is the plant that he actually wanted, this broadleaf one. So if you fertilize plants, all the weeds grow. But if you fertilize them with phosphite, most weeds can't take that up, but these engineered plants can. So this is an example of someone looking at what the environment has and being able to bring that into his own research. And then finally, last year, I met this really remarkable woman at a meeting who, so Rana Dajani, she's a professor in Jordan. She had been doing a postdoc in the United States in immunology, wanted to return to her home country, said, I don't have the resources to do the kind of immunology that I was doing before. What can I do that's both meaningful and so intellectually stimulating and meaningful for the population? So her case, she switched into human population genetics and GWAS studies, and realized that there were a number of other countries who had taken their populations and said, we can learn, because of the history of those populations, you could learn certain things about genetic diseases. And so she looked at two populations in Jordan, Chechen and Circassian populations, and actually used that as a way of developing GWAS studies to model diseases that are important for people in the Middle East. And so I thought that was a really inspiring way of saying this is something that she looked at, something that was valuable to her country as well as unique. So, and so then the final thing that I wanted to just add was that the work that everyone does is in collaboration with other people. And as you're starting your own groups, that's going to be both a huge source of stress, but hopefully also very rewarding. And I've been very fortunate in having a lab that's really from all over the world, coming with all different kinds of expertise. And the thing that's been really freeing for me, I went from being a postdoc, being fairly autonomous, starting my own job and feeling very like I didn't know enough and that I had to be the top of everything. And over time realized that everyone feels like a little bit nervous and wanting to have an expertise in something. And that I could actually relax a little bit. I didn't have to be the smartest person in the room. And in fact, if I let other people shine, they both took responsibility for being, here's our expert in cell cycle, here's our expert in proteomics. They could take that over. They had skills that I didn't have. And as long as we all had some expertise in something and some pride in that, we were also able to ask each other help for the things that we didn't know about. And so that became a really useful thing. And so that is my lab as of a few months ago. There's actually a couple of people that have changed already, but this group has been fantastic, as have the other ones. So I'm going to stop there. Thank you for listening, and I'm happy to take questions.